you. Aroskar sir, Dr. Vinay Aroskar, can you hear?
Yes. Now recording is started, sir.
Yeah.
So shall we start the lecture, Patankar sir?
Yeah, we can start.
Okay.
Hello, Ranjit Gurau, Dr. Ranjit Gurau. I am Dr. Patankar. Hello, can you hear?
Yeah, I can hear.
Yeah.
We can hear.
So I welcome you in this meeting. I am a national convener of Microbiology Society India. So I welcome you on behalf of MBSI, and it's our pleasure. I have seen your topic. It is very interesting, and definitely all participants will enjoy about your topic, and also you please guide the students particularly that how you have reached to states and at your present position. So there are other persons are also there. Dr. Vinay Aroskar, he is a fermentation microbiologist, or you can say the industrial biotechnologist. Dr. Aroskar, can you hear me?
Yes, sir.
Yeah.
Yes, sir.
So, so he is a very eminent person from the industry and working in the Fermenta Biotech. Then Anand Joshi, please introduce yourself. Anand Joshi. Hello. Anand Joshi is there. Suman Chaudhary. Suman Chaudhary.
Yes, sir.
Yeah, please introduce.
Sir, myself Suman Chaudhary from Maharaja Ganga Singh University, Bikaner, Rajasthan.
Oh.
I am pursuing MSc in microbiology.
Very good, very good. Tanvi Raut.
Hello.
Tanvi Raut.
Yes, sir.
Yeah. Tanvi Raut.
Myself, Tanvi Raut.
You are from where?
I am from Nagpur, sir.
Nagpur. You are from Nagpur?
Yes, sir.
From which college?
Congress Nagar Science College.
Okay. From Pranita
Pursued MSc from microbiology.
Okay. Vinita, you are from which college?
CTT college, sir.
Huh?
CTT college, sir.
Okay. So we will proceed. Pranita,
Yes, sir.
you can proceed.
Hello.
Okay.
Yeah.
So on the occasion of 124th birth anniversary of Dr. Panchabra alias Bhausaheb Deshmukh, on behalf of the organizing committee of Honorable Principal Professor MP Dhore sir of Science College, Congress Nagar, Nagpur, and Honorable President, Microbiologist Society India, as well as National Coordinator Honorable Dr. S.D. Patankar sir, on behalf of the organizing committee, I welcome the eminent guest speaker of today's guest lecture, Dr. Ranjit Gurau sir from Texas State University, USA. So before to start the lecture, I must introduce our eminent guest speaker, and he is speaking on the very good topic that microbial fuel cell, a new technology for harnessing electricity from waste using microbes as the biocatalyst. So I take this opportunity to introduce our eminent guest speaker of today's guest lecture series, which is based on the theme Indians Speaking from Abroad. Dr. Ranjit Gurau sir is presently working as a postdoctoral associate scientist at Ingram School of Engineering, Texas State University, Texas, USA. And recently, yes, it is a very proud moment, and it is our honor to share with you all that our Dr. Gurau sir is listed among top 2% scientists worldwide on the basis of his Scopus 2022 data, which is published by Stanford University of USA. So I think it is a big round of applause for
Congratulations.
Dr. Gurau sir. Yes, I have.
Congratulations.
Yes, congratulations, sir. And really, it is our honor that you have accepted our invitation to deliver your precious talk in our lecture series. And sir, it is very great that I have seen your news in the newspaper also, and I feel very proud that I am connected with you. As per the educational qualifications, he has pursued his BSc from Shivaji University, Kolhapur in 2007. His MSc in biotechnology from Shivaji University, Kolhapur in 2009, and PhD in biotechnology in 2013 from the same university. In his research experience, he is the postdoctoral associate at Ingram School of Engineering, Texas State University, San Marcos, Texas, USA, from 2020 to September to till date. Before that, he was a researcher at Department of Biological Engineering, Konkuk University, Seoul, South Korea, from 2021 March to August 2022. He was assistant professor from 2018 to 2021 at Department of Biological Engineering, Seoul, South Korea. He was a principal investigator and worked as a young scientist in the Fast Track Young Scientist Scheme funded by DST SCRB New Delhi, India in 2015 to 2018 from Department of Biotechnology, Shivaji University, Kolhapur. He was a postdoctoral researcher at Department of Environmental Engineering, Nankai University, Tianjin, China. He is the recipient of many prestigious awards and honors. He has received KU Brain Pool Research Fellow Award from Seoul, South Korea. He has received a grant from SERB as a young scientist from SERB at New Delhi, India. Senior Research Fellow Extended Award, CSIR SRF Extended Award from New Delhi, India. Senior Research Fellow Award again from the New Delhi, India, from Council of Scientific and Industrial Research, as well as Basic Scientific Research Fellow and Departmental Research Fellow Award. He has his expertise area in environmental biotechnology. He has worked on the remediation of petroleum, hydrocarbons, textile dyes, antibiotics, plastics, and pesticides, as well as heavy metals. In microbial fuel cell technology, he has worked on the isolation of electricogens for the electricity generation and utilization of waste biomass or waste water. In biochemicals production, he has worked on production of amino acid cocktail and acetyl glucosamine, biodiesel, etc. In biopolymer production, he has worked on bioplastics and exopolysaccharide production. In enzymology, he has worked on chitinase and keratinase production, separation, and its purification. In molecular biology, he has worked on isolation of DNA, RNA, PCR amplification, and RT-PCR analysis of sequencing data, etc. In toxicology, he has worked on assessment of cytotoxicity, genotoxicity, phytotoxicity, and stress-related enzymes. In nutrition, he is expertise in antioxidants and biochemical analysis of agricultural products and its byproducts for nutritional factors. In soil analysis also, he has worked on the soil metagenome, contaminants, salinity, micro and macro elements. And in analytical section, he has worked on MFC, HPLC, as well as GCMS, GCT-CD, RT-PCR, etc., etc. He has received a grant from Science and Engineering Technology Board, New Delhi, in 2000. I think on the topic entitled Exploring the Novel Chitinase Using Metagenomics Approaches and Their Anti-Phytopathogenic Potential, which is costing rupees 30 lakhs. He was the principal investigator there. He has more than 96 international publications. He has the total impact factor of Scopus is more than 500. He has 2 patents to his credit. He has book chapters published in CRC Press, Elsevier, as well as Springers. He has very good citation for the research articles that in the Scopus Index journals, he has the citations of 1813, as well as H index is 24. In the Google Scholar citation, he has overall the citations of 2199, as well as H index is 26 and I-10 index is 49. He has very good publications in various reputed journals, as well as he is the editorial board member of Frontiers in Microbiology, which has the impact factor of 6.064, as well as he is the editorial board member of Research Journal of Life Sciences, Bioinformatics, Pharmaceuticals, and Chemical Sciences. He is the reviewer for the international journals to his credit, and he has the membership of various professional bodies like Biotech Research Society of India, as well as Association of Microbiologists Society of India. So thank you very much, sir, and really a very good round of applause for you, sir, and congratulations. And as well as thank you on behalf of the organizing committee that you have accepted our invitation. So it is a request from on behalf of the organizing committee that please deliver your precious lecture. And before that, please elaborate your journey from India to abroad. Please.
Okay. Thank you very much for the introduction. So I'm clearly audible, right?
Yes.
Okay. So myself, as already you have introduced me in detail. So I think so there is no more detailed introduction needed. But at the end, I think so I will first cover my topic, and then we can discuss about the journey and all these things. So I will just first cover my topic.
Okay.
Share my. Okay, so
yes,
I think so my screen is visible to everyone.
Yes.
So although it's morning over there and it's night in Texas, so it's now the time is like 10:50. So today is Saturday here. So a very good morning to all of you, according to Indian time, I can say. So today I will be talking about the microbial fuel cell, a new technology for harnessing electricity from waste using microbes as the biocatalyst. So presently I'm working in Ingram School of Engineering, Texas State University. So as we know that energy, so energy is the one term that is widely used for different kind of application, or it's highly needed that we need energy for different purposes. So usually for energy production or any consumption is we have liquid fuels that are 33% used, that are like oil, we have petroleum, gasoline, and everything. Natural gas is 23%, coal, then nuclear and renewable energy. So only 12% of the renewable energy is used. So basically all these energy sources are widely used for industrial purposes, transportation, residential area, commercialization. So basically industrial and transportation is mostly they consume a lot of energy. So this is a kind of graphical. We can say that it's a predicted, or it's a statistical data that where we can see that this is the nuclear power. So this is the 2050 and 2030. So nuclear power will be not more utilized till 2050, and then renewable sources, petroleum, natural gas, and coal. Here we can see that most of them are till now at the highest point, petroleum and other liquid fuel are used, and they will be continued to use till 2050. But however, renewable kind of energy that it has been started like to grow day by day, and in 2050 it is predicted that petroleum it can take over to the petroleum in the best any kind of fuel or natural gas and all these things. So at 2050 it will be around 28% of the renewable energy share will be in the market. So right now there are several sources of renewable energy use. That is the biomass based wave. We have the seashore. So it's a kind of we can say that wave energy. Then solar is widely used in India and everywhere. then the water, and then geothermal, and then wind energy. However, we also depend on the non-renewable energy. As I said, that nuclear, coal, and then the oil and natural gas are the most widely used renewable sources, non-renewable sources. But the time has come that we have to think for the alternatives, that as the renewable, non-renewable energy sources are, one day they are going to be empty, because there will be no more digging and we won't get any oil or any gas. So we have to start thinking about different way that how this renewable energy can be used. So although these like solar energy and wind energy and water energy are used for a long time, but this is a kind of we can see that how it will turn renewable energy goals. So most of them and most of the energy part will be covered by renewable and sustainable energy in 2050. However, that each technology has its own pros and cons. So there are some pros and cons of the renewable energy. So like pros, like good things are that it won't run out, because it's a renewable energy, so it won't run out. That we have plenty of wind, always it will be available, sunlight, water will be available. So, and even the biomass will be available. But so it is a plenty of energy we have, researchers we have. It has a numerous health benefit, that as a other non-renewable can release the toxic gases and a lot of pollution. So another is that a low reliance on foreign countries. Now we depend on the Dubai or any Saudi countries for the oil, and Russia. So there is no need to depend on any other countries. But there are also some cons like intermediate. So sometimes there won't be more sun, or there won't be a water more that we can use to flow from the dam. That so if there is a drought or anything of condition, or if there is a in other countries like there is a snowfall, so that covers the that can cover the other thing. So another is like need of energy storage. We have and geographic limitations, climatic, weather, and everything. So plant-based energy, as we know that biofuel, biodiesel has been widely used, or it has been widely produced. So this is the first generation that is the edible biomass we use, but this is not any more accepted now, because there is a food scarcity. So usually we don't prefer to use the first generation fuels. So we have to think about the second generation as it is the kind of waste. So it is we have the wood, straw, grass, waste. So we can use the second generation fuel. Whereas the third is the algal, and then another is that pyrolysis of the biomass, and then we can use that. So we have now the second generation that already people are using. So can we use this for other purposes? Like already the second generation is used for biodiesel production, but can we use it for any other purposes? So the idea behind this was to produce the electricity using waste biomass from the lignocellulose biomass or any kind of waste biomass. So we have recently we have the technology that is called as microbial fuel cell. So what is this microbial fuel cell? So it's a kind of electrochemical device that harnesses the natural metabolism of microbes to produce electrical power directly from organic material, any organic material. It can be any kind of waste, or it can be any substrate. So basically, it's a kind of chemical energy to electric energy conversion using microbes as the catalyst. So before moving toward the microbial fuel cell, let us discuss about the dry cell. We have everyone has seen the battery. we have the torch or we have the any kind of battery cell in our house. So basically, this is the structure of the battery cell, that it has the cathode and it has the anode, and from then electrons will transfer from the anode to cathode, and we can glow the bulb, or we can use it for lighting purposes and different purposes. So this is the kind of normal normal cell, dry cell. It's a chemical cell. It's not a microbial cell. So it has the similar mechanism. Why I showed? Because the microbial fuel cell will have the similar kind of mechanism. So microbial fuel cell. Now this is the microbial fuel cell. So basically, as I said, that there is an anode compartment here, and then there is a cathode compartment here. And then usually the anode compartment will be aerobic. There will be no any kind of oxygen, or it should be have limited oxygen supply. So usually at anode, we cultivate the bacteria. So this is the kind of two-chamber microbial fuel cell. So the idea behind this is that this is the electrode. So we have the microbes, and then we can add any kind of nutrients, and then electrons will be transferred from anode to cathode. And then this is the proton exchange membrane. So it can exchange the protons from anode to cathode. So detailed explanation I will show in the next slide. So basically, microbial fuel cell is the one kind of cell, but there are different kind of electrochemical cell, depends on the application. So it's a microbial carbon capture, desalination cell, electrolysis cell, then photosynthesis, methanogenesis, remediation, and different cells. So we will focus on microbial fuel cell. Our today's topic is microbial fuel cell. So we will discuss on that. So these are the types of MFC, that is microbial fuel cell. So they are the stacked one, they are the mediator dependent, mediator-less, and thermophilic, mesophilic, and then single chamber, dual chamber, flow up. So these are the different kind of cell. Now, why we what is our interest? We want to remediate the pollutant, or we want to produce the electricity, or it depends on the application and capacity of the cell. We can choose. So these are the few examples of the microbial fuel cell that are widely used, that are widely used in the lab scale, or they are being started for commercialization. So basically, this is the two chambers fuel cell. So this will be anode, and this will be cathode, and then the electrons will flow from here to there, and then this is the membrane. As I said, that proton exchange membrane. So these are the different like tube microbial fuel cell, stacked microbial fuel cell, cassette microbial fuel cell, and there are different. So basically, many people are testing different kind of microbial fuel cell to enhance the power production, or to decrease the resistance, or to test the which electrode is best. And there are so many things that they use that. So this, as I said, that microbial fuel cell has several applications. So basically, this is for generation of so this is basically for electricity generation, what we will be talking on. Then we will be also talking on bioremediation of microbial cell, biosensors to detect any kind of pollutant in the water. Then there are several applications of the microbial fuel cell. I think so. My screen is freezing. Okay, so there are different kind of substrate. As microbes need a kind of feed, or it need a fuel for itself to break down that fuel, and it can be used for microbial growth and for energy production. So there are different kind of substrate used. So these are the non-fermentable sugar substrate. So these are acetic acid we can use, acetate, butyrate. These are different kind of organic acid we can use. Then fermentable and complex substrate, we can use glucose, cellulose, maltose, saccharose, cellobiose. And also we can use as the wastewater, like domestic wastewater, livestock, refinery, food processing, and different kind of wastewater as a feedstock for microbes. But as I said that we are here trying to develop something sustainable, so it should be cost effective and it should be used some kind of waste. So basically the idea behind this is to utilize the lignocellulose or any kind of plant derived waste matter that as a substrate. As we know that lignocellulose biomass is plenty, it is available in plenty. So it's from the agriculture, it is from the industrial and different areas, or even we get that kind of waste in forest waste. So, but the problem with this waste is like we cannot apply lignocellulose leaves or roots or any kind of plant material directly to the microbes, because few microbes have that system, but it will take a long time to break down this complex structure and release few sugar that can be used by the microbe. So basically, there are some pre-treatment methods used. So this is the plant biomass, and we know that it utilizes CO2, light, and it can enhance the biomass. So we have the plant biomass, and then these are the different acid or base hydrolysis method, or we can use microwave or ultrasonication, or there are some enzymatic method to hydrolyze this complex plant biomass into monosaccharides or oligosaccharides, like, for example, glucose and xylose are the main product of hydrolysis. So we need that product to feed our microbes. But the drawback of this system like acid-base hydrolysis or this, they don't only generate the monosaccharides like glucose or xylose, but they also produce some kind of toxic chemicals or intermediate. For example, like 5-hydroxymethylfurfuryl limonenic acid. There are several phenolics. In next slide, I will talk about that. And so these monosaccharides can be fed to the microbes, and then we can either produce gas, we can produce a biohydrogen, or a liquid fuel, that ethanol, butanol, or our target is to use it for microbial fuel cell, and it can be used for other purposes. But as I said that there are some kind of toxic intermediate. So what if these intermediates are present and they inhibit the bacterial growth? So that is not a plus point. So there are kind of furfuraldehydes. This is our widely present hydroxymethylfurfuryl, furfuryl, acetic acid, some organic acid, phenolics, valine, and all these things. So they usually forms of the degradation of different kind of hexoses, pentoses, hemicellulose, lignin. These are the byproduct of the degradation of the plant biomass. But they can damage the cell wall and DNA, and then they inhibit the growth of microbes. So what people do, people usually use different kind of pre-treatment. Either they genetically modify the bacteria so that it can tolerate the high concentration of these fermentation inhibitors, or second choice is the pre-filtration of the media, activated carbon, biochar, or any kind of pre-treatment is used to remove this kind of intermediate from the fermentation media. But alternatively, this pre-treatment method can enhance the cost. Sometimes they are not very well effective. So we have to think that are there any microbes that can tolerate this kind of inhibitors? So basically, this is the list of different microorganisms. They have been listed or they have been widely studied for the electricity production. So this is like Aeromonas, and there are several microbes. But Geobacter and Schwannella are the most widely studied because they produce the higher amount of electricity. So they are widely used for different purposes in microbial fuel cell. So, however, they have some restriction that, for example, Bacter can only use the acetate, whereas Schwannella can use the lactate, pyruvate, acetate, and glucose. some Schwanella, the higher producing electricity producing Schwanella can use only lactate. Now the problem start here. Although they are the high electricity producing microbes, they cannot utilize the glucose or xylose, or they cannot tolerate any kind of inhibitor. So in that case, most of the people recently they have started genetically modifying the bacteria to either to utilize or metabolic pathways engineering on all these things. So they can utilize glucose. They can also tolerate high concentration of inhibitors. But the drawback of this engineering on all those things is that they also increase the cost. System is not stable, and another thing is that the electricity production is due to activation or shutdown of many pathways. The electricity production is affected. So what we did, we tried to isolate few microbes that can tolerate the inhibitors, that can utilize the glucose and xylose. As we said that Schwanella can use only lactate, pyruvate, or N-acetyl glucosamine, and the cost of that is very, very high. So it's not a sustainable technology, or it's not for long-term goals. So in this case, we have used like barley, miscanthus, and pine. So this was chemically and enzymatic treated, and then we got glucose and xylose, and then it was fed to the microbes. So basically, we isolated the Schwanella morris flavi. So it was isolated from the seawater. So although it was isolated from the seawater, it can tolerate the high concentration of different inhibitors, different metals, and it was able to tolerate any kind of pollutant. So the beauty of this bacteria, it can use glucose, xylose, tolerate high concentration of inhibitors, heavy metals, or any kind of pollutants. So here at the anode chamber, we placed the bacteria, added the glucose or the plant hydrolyzed whatever we have. We have barley, miscanthus, and pine. They were separately fed to the microbes at the anode, and then we set the resistance like different, like we set here for 1001, and then this is the cathode. So whatever plant biomass we have used, so it has a glucose, xylose, so it has a barley, miscanthus, and pine. So we have 135 gram per liter of glucose. Then this is the xylose. The xylose concentration was low, glucose concentration was high. Now this each has a different different composition, because the pre-treatment method or the plant biomass, source of biomass, it changes. And also the concentration of acetate, furfuryl, and hydroxymethyl furfuryl is present, that acts as an inhibitor. So this was the phylogenetic analysis of that bacteria. That's a bacteria is like a sea salt harvesting area, where usually they produce the salt, or they extract the salt from the sea. So this bacteria was isolated from high salt area. So on feeding this kind of different hydrolysate for the bacteria to produce the electricity, so we can see that this is the current density milliampere centimeter square. So we have the electrode. So this is calculated as on time. So this was tested for like for a long time, and then because testing for a long time is very, very important. First thing is that it should last for a long time, because if the media is depleted, then the current density will fall very soon. So first thing was it has a glucose and xylose. So the preference was given to both. Then there are some organic intermediates present. They were also utilized. So electricity production was continued for a long time. So first is this is the barley. Barley hydrolysate give the higher electricity production, followed by miscanthus and then pine. Now you say that why this different is this? So basically this, although the glucose concentration was adjusted, but the inhibitors present in them, they have the different concentration. So this, if they have the high, for example, pine has some kind of phenolics and higher kind of intermediate that were produced during pre-treatment. So it produced the electricity that slowly, and it takes some time. So basically barley produced like 6.66 milliampere per centimeter square was the electricity. This is the miscanthus, and then pine. So pine showed less. So this was the set that the resistance was set to 100 ohm. Glucose was adjusted to 1%, and then we can also feed them with the iron. So okay, so this is very, very important when we work on the microbial fuel cell or any kind of fuel cell, that we need to know that how the microbe can transfer the electrons from microbe to the anode. So, and from anode, how will they travel to cathode and all these things. So this is usually used as like a barley, miscanthus, and pine. So let us see that, for example, we have the peak here at 400. So we have the peak at 400. Also, we have the peak at 200. So what does it determine? Now I will show you in detail. So basically, as we know that when we add glucose or any kind of media or any kind of nutrient to the bacteria, they hydrolyze it, and then TCA cycle, also carbon dioxide is released, and then there are electrons produced over here. So what happens on oxidation of any kind of substrate, any glucose, maybe acetose or xylose, or any kind of wastewater that has glucose, xylose, or any kind of organic contaminant. So what does bacteria oxidize them, and then they utilize as a nutrient, and they release some electrons. Now they release the electrons, but how that electron can transfer from anode to cathode? Because if the bacteria, for example, take this image. So for example, if bacteria is freely in the media and this is not stick to the anode, so how they can transfer the electron? They don't have any mechanism to transfer the electron. So first thing is, like, if the bacteria get attached to the anode, they can directly transfer the electrons from bacterial cell to the anode. So this is called as direct electron transfer. Direct electron transfer means they form a thick biofilm on the anode, and they can easily transfer the electrons from the bacterial cell to the anode. So basically, they have the cytochrome kind of C, cytochrome C, cell-bound cytochrome C, and there are outer membrane bound cytochrome, and then MTR, and there are different kind of direct electron transfer mechanism. So what happens if this is the anode surface and bacteria get stick to the anode? They can easily transfer directly electrons from the bacterial cell to the anode. But in case if some bacteria or the bacteria which has not formed the biofilm and the bacteria is freely in the media, how can they transfer the electrons? So they need a shuttle. For example, if you want to travel from this place to that place, we use the shuttle, like bus. We sit in the bus or any car, and then we move from one spot to another spot. So they need a kind of shuttle. So they need some shuttle. So shuttle are different intermediates. They are flavins, they are quinones, they are soluble metals. So these are like quinones and flavins. They are produced by microbes. So they act as the shuttle. So they capture the electrons from the bacteria. They go to the anode, they release the media, then they go back, then capture the electrons, and then this cycle, this shuttle keeps on continue. So this is the where here we can say that it is bound. Cell is bound to the anode, and it can directly outer cytochrome, they can transfer the electron. However, some bacteria like Geobacter has a nanowires. They form the network like wire. They connect each other using their wire network. So they, in that case, they can transfer the electrons through the wire to the anode. So these are all direct because they are connected to the anode. So that is direct electron. And then in indirect or mediated, that means it needs some mediator to transfer the electron. So here we can say that this is reductive and oxidative reaction happens, and then they keep on transferring the electrons from anode. We can see that how the electrons transfer from anode to cathode. So as I said that this is the insoluble electron acceptor. This is means as I said that if they have the nanowires and they can transfer the electrons directly, but these are some soluble electron acceptor. Here we can say that this is the shuttle. So electrons directly pass from the outer membrane freely to the media, and then some mediators like, as I said, riboflavin or quinones or any soluble metal, they capture the electrons and then they transfer to the electrode. So this is the down image. We can see that this is the Geobacter. So see how it has a network, wire network. So this is the nanowires. Okay, so what are the reaction? If what will be the final product or how the reaction occurs? Now we have the anode and we have the cathode. So what are the reaction at anode? Because at anode, we will either fill with the glucose, or if we fill with the acetic acid or acetate. For example, if we fill with the acetic acid, then water is the first acetic acid plus water. They will give 2 molecules of carbon dioxide and then 7 protons plus 8 electrons. Now, as I said that this is the product at anode. So anode has 7 protons when we use the acetic acid, and it has 8 electrons. Now what will happen? That 8 electrons will transfer from anode to cathode using the external circuit. And how this proton, so we have the proton exchange membrane. So this proton will transfer from anode to cathode through the proton exchange membrane. So this is the kind of anodic reaction. In case if we use the glucose, we get a higher number of protons and electrons. That is 24 protons and 24 electrons we get if we use glucose. So now our electron has been transferred from anode to cathode, and our proton has been transferred from through membrane to anode, from anode to cathode. So now what will be the cathodic reaction? Because as I said that anodic chamber will be anaerobic and cathodic chamber will be aerobic. So what happened at the cathode? There is a oxygen. As it is aerobic, we have the oxygen, and then four proton that has been. If we use the 4 proton, it needs 4 electrons, and then final product will be the water. So in all cases, final product is the water. Now, if we want to produce hydrogen gas or any kind, we can use different kind of electrodes at the cathode, and we need to make that chamber anaerobic. So it depends on our interest or our product interest. So coming to our topic, that we have barley, miscanthus, and pine. So I think so this, it has been overlap. I cannot see this, but maybe at this place we can see that barley showed the higher power density, followed by miscanthus and pine. So as a TCA cycle has different intermediates. Now, at the first point, I talked that we need to produce electricity for a long time. So, for example, glucose is exhausted or xylose is exhausted. How microbes can survive? Whether they can use any kind of intermediate that were produced during the TCA cycle. So these are the different organic acid that were produced. So we can see that Barley hydrolysis produce more organic. So these are the fermented. I mean, these are not produced in the pre-treatment. These are produced by the bacteria during utilization of glucose or xylose. These are the intermediate products of the TCA cycle or so metabolic cycle. So they produce like pyruvate, succinate, lactate, formate, acetate, and now we know that lactate is usually used by the Schwannella. Formate is also used, and acetate is also used. So here we can see that at 48 hours, or 96 hours, they produce higher amount of organic acid, and then the concentration was decreased. That means initially glucose was of the interest of the bacteria, so usually they try to utilize the glucose, and then on depletion of glucose, whatever organic acid were present, they started, or it is the chance that they have simultaneously started utilizing. So we can see that the concentration of this organic acid was decreased in every case. Fine shows low current electricity production, and also it shows low organic acid production. This could be that bacteria could not grow very well. So that could be the chance. So this is again using the barley, miscanthus, and pine. So you can clearly see here that how these bacteria get settled on. So this is the anode carbon electrode. So this is the carbon electrode, and the bacteria has very well formed a biofilm on the electrode. You can clearly see that. So that means, as I said in first part, that the Schwannella we have isolated have, I can show that. Okay, so Schwannella we have isolated. It shows the peak, like this is the cyclic voltammetry. It is used to determine the electron transfer mechanism. So for example, if we have the peak at 0.4, so that means it has the produce some kind of flavins. So that flavins, like, they help to transfer the electron. So these bacteria, it produces some kind of flavin or any kind of external mediators. So they transfer this electron from media to the anode. But this bacteria also has, we can see that there is a peak at 200, 0.22. So that means it has also the capacity to transfer the electrons directly from the outer cell membrane to anode. So it has two mechanisms. This bacteria has two mechanisms. It can produce, I detected, and it was it produces riboflavin. So this bacteria can produce riboflavin that act as the external redox mediator that can transfer electrons from the media. So it is the shuttle. And another mechanism that it can attach to the cell and to the anode, and it can directly transfer the electrons from outer cell like cytochrome C or outer membrane bound cytochromes to the electrode. So here we can see that it has formed a thick biofilm. So first thing is that it needs a support. So it could have taken the support of the electrode to settle, but we have already done the cyclic voltammetry. So cyclic voltammetry data said that whatever bacteria has found the biofilm on the anode, they have the ability to transfer electron directly from the cell to the anode. So this is the biofilm. We were not able to see any kind of nanowires because it needs some kind of special tools, or it may be present or it may be not present. Okay, so in first part, we utilize, so I will summarize, we use miscanthus, pine, and then we use barley straw. It's not a barley, so it's a barley straw. So we hydrolyze it, use it as a media, and then bacteria produce some electricity. Now that was about the lignocellulose biomass. Now second biomass is like the chitin or chitin we call is that. So whenever you go to any kind of market or any chitin resources, like we have the shrimp waste, we have the chitin waste from the crab. People eat the crab, but they don't eat the shell or the outer shell. So it is the disposed or it is waste. Some people use it for different purposes, like they use for preparing some. They use to prepare some colloidal chitin or the chitosan or different applications. So what I did, I used shrimp and crab and isolated some kind of, this is the pre-treated chitin, and then we have the chitin biomass, and then I use this to feed to the microbe at the anodic chamber. So this bacteria was different. So I use two new isolated. They can tolerate the high salt concentration. So what was the idea that as crab and shrimp is widely used or used in the seafood industry, so it has a lot of brine. Brine means high salt content for preservation and all these things. So if the bacteria can tolerate this high brine, it is a good opportunity or good possibility that we can use this chitin in a microbial fuel cell that has seawater as a nutrient media. So in this case, what I did, I used the seawater and chitin, only chitin plus seawater or chitin and seawater as a source of nutrient, and this bacteria can hydrolyze and produce some electricity. But then like the story was slightly changed, and then the one aim was to produce the electricity. Then how about, because after degradation we have some byproducts like N-acetyl glucosamine has a lot of applications. So it produces some, after hydrolysis of chitin, it produces N-acetyl glucosamine, and that has a lot of medical application and so on. So first aim was to utilize chitin in the media, and then after hydrolysis, first product will be the electricity. Second product will be the N-acetyl glucosamine that is freely present in the media. And third product will be bioplastic. But as I said that the strain, whatever I have isolated, they were not able to produce the bioplastic. So what I did, I used the consortia and utilized the Cuprividis necator. We also call these as a Ralstonia eutropha. So Ralstonia eutropha don't have the system to hydrolyze the chitin. So what they do, they utilize the N-acetyl glucosamine. So in that case, I used N-acetyl like Ralstonia and electricity producing microbes together. So Ralstonia utilize some of the N-acetyl glucosamine, and then they produce the pH, or we can call it as a polyhydroxy alkanite. So there were three products from this system. So first is the electricity, bioplastic, and N-acetyl glucosamine. So here we can see in this image, this is the chitin in the media control, and after degradation, it was completely dissolved and there was no chitin in the media. And this is the another bacteria, as I said, that sea salt harvesting pond, that this bacteria was isolated from that, and only in the growth media, only seawater and chitin was used, no any other source of nutrient was used, and it produces a good amount of electricity. Okay, so the in first case we use lignocellulose biomass, and then we use the chitin. But if I want to treat any kind of pollutant, so I work on also on the bisphenol A, but I will tell you an example here is of the dye, because textile dyes are widely used in India and everywhere, and different technologies are used like adsorption technology or degradation and bioremediation. The problem with this bioremediation technology, solely if you want to use the bioremediation technology, sometimes that is not effective, because if the water or the effluent has the azo dye, it needs some external electrons for cleavage of the azo bond. So in that case, if bacteria, what if we use dye in the microbial fuel cell. They can whether utilize the electrons to or dye hydrolysis efficiency can be encouraged. So I use the like bioelectrochemical system. I name it as a bioelectrochemical system because my aim was not to produce, in this case, the electricity, but to hydrolyze the dye or break down the dye. So in this space, here we can say anodic chamber, cathodic, then this is the second, this is the anodic cathodic chamber. So here we can see that in first VS, I added dye plus bacteria at anode. In cathodic chamber, I added dye at the cathode. And at anode, there was only my bacteria Schwannella. This is also the same bacteria that was used in the plant biomass utilization for the electricity production. So this is the same bacteria. So what happened in first case, I will talk that we can see that there is a dye and there is a bacteria together at anode. So dye and bacteria, bacteria produces some electrons, but dye wants the electron, but anode competes with the dye and they take the electrons. So electrons are not available for the dye. So in this first case, dye was not degraded. We can see here, this is after 48 hours. But in second case, what is the second case? We have the anode and cathode. So bacteria was cultivated at anode and also at cathode, but cathode has the dye. So in this case, at anode, bacteria produce some electrons transferred to the cathode. So at that point, instead of being a water as the final product, dye utilize that release the electrons and they break the azo bond. And so dye decoloration was observed. We can see here after 48 hours, complete decoloration. Whereas in third system, this is the control system, no bacteria, only dye at the cathode, whether it can be degraded or not. Okay, so so this is the 97 decoloration, 84 and percent decoloration was observed. Okay, so moving towards actual utilization of microbial fuel cell, whether we can utilize it for some other purposes or this system can be applied or combined with the plant system. As I said that they need a continuous supply of the nutrients. So how if we combine this microbial fuel cell technology with the plants? So yeah, as we know that plants grow and they release some nutrients in the root zonal area. And so rhizospheric microbes, usually they grow at the root zone or rhizospheric zone, and they utilize the whatever nutrients that are exuded from the roots, they can utilize it for their growth. But in this case, how about if we use this nutrients released from the plant to grow our electricity producing microbes? So this is basically if in a pond at upper layer, if we have a cathode, then because cathode, if it is exposed to air, it doesn't matter. So for example, if we have the wetland on top floor, there will be cathode, and below under the soil, there will be the anode. So whatever roots will release the nutrients that can be utilized by bacteria, and they can produce some electricity. So there is no need to supply any additional nutrients for this. Plant can provide some kind of waste exudate and bacteria can grow. So this is basically it can be used also for the effluent, because if we have this kind of wetlands or pond or any kind of water that, any kind of water plants that grow in the water. So here we can see that this is the cathode and anode placed at the bottom, and then we can transfer the effluent from down. And so it will first come in contact with the anode. So whatever soil microbes are present, they can also utilize wastewater or effluent or municipal, agriculture, industrial, dairy, different kind of waste effluent. And then it can come first in contact with the anode. So whatever bacteria grow at the anode, if they are able to produce any kind of electricity, they will stuck or they will form a biofilm at anode, and then we can see that the effluent, this is the clean water. This is the kind of a prototype used in the for wastewater treatment. Okay, so this is the lab scale microbial fuel cell. Sorry, plant microbial fuel cell. We can call it as a plant microbial fuel cell because plants are present, because we have the plant can fix the they can take the sunlight and fix the atmospheric carbon dioxide, and they can produce their own energy. So microbes can depend on plant, but plant, there is no need to depend on any kind of additional nutrient. If there is some wetland plants are there, they can utilize some waste from the plant for their growth. Okay, so this is the first commercialized, the Dutch company, they call this as Plant E. For example, we call this E. E is for electronics or any kind of this. So they have produced plants that can produce the electricity. So this is they can light up some LEDs and all these things. So possible plant-based system application where it is possible. So these are the different countries. So for example, in US, green rooftop is possible. In India, we have the rice field, the wetlands. So it is possible to apply this technology. So on rooftop in US some part, and then for example, if you want to apply it for in India, we have a lot of rice field in south part, so it can be used. So this is the first commercialized E plant. So this plant can produce electricity for 24 hours because bacteria won't stop to produce electricity until they get any kind of if there is a limitation of any kind of nutrients, or if the plant dies, then that is the only chance. Otherwise, it can just keep producing electricity. So this is the first commercialized product, planting. So it has been used in some as a decorative. Still, at a higher scale, it is not possible, but as a decorative and for in the pot scale, they have succeeded. So this, as I said, on rooftop we can use, and then we can use for pollutant degradation and different purposes. So this is the clean energy, renewable energy, day and night it can be produced, and then it is the efficient. No need to harvest or no transportation is needed, and then nutrient removal or nutrient preservation and water treatment. Okay, so this is my earlier team in China, and this is from South Korea, and now this is in US. So we have a good team. So if you have any questions, please. Thank you.
Thank you very much, sir, for your thorough information on the generation of electricity by using the fuel cells. That bacteria plays very important role in electricity, and it is the need of time in future perspectives point of view, that we could use the renewable sources of energy, so that we could not means we should avoid the difficulties of the fuel in future. So it is very great, sir. So if the participants have any queries, I can type in the chat box.
Yes, sir.
Yeah.
We can hear you.
I congratulate you for such a nice lecture. It's a very informative one. My interest it is in have you scaled up the dye degradation process by using this technology?
Actually, we have not scaled up, but we have done it up till 2 liters.
Okay.
So this reaction is actually specifically if we have a azo kind of dye.
Yeah.
If we have a microbial fuel cell system, we call this as a bioelectrochemical system because we don't want to generate any fuel.
Yeah.
So our aim is to degrade any kind of product. So yes, it can be scaled up to it depends on size of our electrode we have.
Yeah.
So if we have a big size of electrode, or we have a big kind of a simple drum, plastic drum can be used,
Mhm.
because it will not produce a lot of gases, or there is no
Yeah.
chance to burst. So simple, if we have two kind of drums, and there should be a connected pipe in between, and then and membrane, and then just a external connection.
Circuit will be given. So which organism you have used there?
I use the Schwannella maris flavi, because that has the ability, as I said, that it can use a glucose, xylose, any kind of waste contaminant, it can be used. So of course we can use any other microbes, but the problem is that if they don't have any ability to transfer the electrons,
Yeah.
so in that case, then if it has the higher ability to transfer the electron, there is a more chances of easy degradation of any organic pollutant.
Yeah. Thank you. Nice one.
There is one question in the chat box.
Yes.
When we can scale up commercially?
You mean remediation or electricity generation? Scale up.
The question from Vinay Aroskar, sir.
Electricity generation.
Okay, so for scale up, many people actually the lab scale fuel cell, for example, we have anode and cathode. They cannot be utilized or scale up for long. But if it is a plant microbial fuel cell, we have a rice field. For example, if we have a rice field, we can just place the cathode anode at the base. So yes, it is possible that we can scale up this process in upcoming days. But people are working on the cost. Cost is very important. Cost of the microbial fuel cell, the most cost part is the electrode. So people are trying to utilize different kind of like carbon electrode, graphene, or any inexpensive method or any inexpensive material that can be used as anode and cathode. Because the problem of the cost start at this material. Microbes, there is no need to, as I said, they can feed effluent, they can use the whatever exudats that are released by the plants. So nutrients and all these things are not the cost effective, but the system, the anode or cathode is very, very expensive. So people are trying to work more on that. So we have also tried to simple like a metal plate, like iron metal plate, and then we try to coat it with some kind of other material. Because if we use iron, then the problem is that rusting. It easily get rust, and then it get easily the system or the net electricity connection easily get collapsed. So they need some coating that the iron should not easily degrade, or it should not rust. So yes, within a Planty company has already commercialized. So they have also commercialized in city. There is a middle path, or the divider we called. So on that divider in Dutch, in 2-3 cities, they have already implanted this microbial fuel cell in the divider, and yes, they produce electricity.
Okay.
So good thing about this is at the remote, if we built a technology, small technology, and if we could at least glow a small room where there is no electricity in any area, so these people will not buy unless that the cost is very low, or any kind of. So yes, any technology needs some time, but as I said, that we don't have choice. In 2050, we will be solely dependent on renewable energy, because at that time we don't have any resources, petroleum or any other.
So there is a question from whether mutation has been tried to enhance electricity generation.
Yes, electricity generation mutation has been tried, but as I said, that the system like as all our microbiologists, we know that if we do any genetically modification, first phase or first subculture, second, third, fourth, it works. But after that, the mutation or it gets reversed or plasmid is lost or anything can happen. So people has tried, but they have failed. So instead of using that system, people are trying to isolate some kind of native microbes that can tolerate or they can produce the higher amount of electricity. So basically, if we have a one cassette or small cell, it will produce low. But if we assemble them as a cassette, that multiple microbial fuel cells, so collectively they will produce a good amount of electricity. So this system is possible for plant-based systems or wastewater treatment.
Okay. So the individual culture means the natural organism is very much beneficial instead of gene manipulations.
I said that gene manipulation is okay, but again, the natural microbes can better compete with others because
Yes.
there is that is their environment. We
Yes,
are not
exactly.
introduce any other guys. They have their own comfort zone that I am comfortable in this room. I don't want any other guy. So they are comfortable. They have their own comfort zone. So they are helping each other. So genetically engineering bacteria sometimes they fail to grow. So it's better always if you want to use on the field study or at the large scale, the native microbes like high salt tolerance. Other microbes cannot grow, but this microbe, if we want to treat any kind of chitin waste, industry waste, it has a high brine. These bacteria can easily utilize the chitin from the brine waste or brine water.
Can we use multiple cultures? Means mixed culture is more beneficial, or we can use the specific one, means the substrate specific organisms only.
Okay, so if we are using the pure substrate, if we are using glucose and we want to test our system and we don't want any contamination, so at that case we can use single microbe. But if there is a consortia, as I said, that there are some intermediate produced by the bacteria or intermediate metabolites are produced. So if the bacteria is able to utilize only glucose and it is not utilizing xylose, but we have other strain that is also electricity producing, it can use the xylose on TCA cycle intermediate as a nutrient source. So consortia can work very well also.
Okay. Yes, sir. So I think if there are now the questions, yes, so it's okay. So thank you very much, sir, for your very nice presentation. Once again, congratulations to you for your great achievement recently that you have ranked in first 2% scientists. And yes, on behalf of the organizing committees, Honorable Principal, Science College, Congress Nagar, Nagpur, Professor MP Dhore, sir, and Honorable President of Microbiology Society India, Dr. A.M. Deshmukh, sir. On behalf of the organizing committee, I am very much thankful to you, Dr. Ranjit Gurav, sir, for your great presentation on the said topic, which is based on the theme Indians speaking from abroad. So thank you very much.
Thing, please give email ID in the chat box.
Okay, I will just give.
Yeah, so someone want to contact you, may contact.
Another thing is that due to time limitation, maybe so if anyone want to do their postdoc, if anyone want to do their PhD, or if anyone want to do their MS, and in different countries, like I have experience of China, Korea, and now US. So if anyone is interested to come in any of these countries, and what are the steps, how to select the professor, what are the funding, and then how to approach, how to write email, and all these things, I have a separate presentation. If anyone want that, I can just email me, I can share, or you can directly email that you got an offer or you want to apply here. So in that case, I'll be there to help, because talking about one thing that because this is a different audience, that we have master student, we have PhD, or maybe we have a postdoc, or so even there are some faculty members, they want to go as an exchange or on sabbatical leave, or as an exchange researcher. There are also several schemes for in that case. So I think I'll share my email, and if anyone has questions, please feel free to.
Thank you very much, sir.
How can you
I
share
hope,
important slides? Hello, Dr. Ranjit.
yes, sir.
Dr. Ranjit,
Yes, sir.
how can you share some important slides
Which
from
slide
your
means?
this presentation?
Okay.
Yes, it will be circulated amongst the groups
Okay.
from the organizing committee later on.
Okay.
And
I will.
Okay.
streaming also.
I will prepare the PDF. So
Yes,
it will
yes.
be small size, and then
It will be.
easily it can be circulated.
Okay.
Yeah,
Thank you
yeah,
so much, sir,
that's
for a
good.
nice presentation. Sir, can you please share your email ID?
Yeah, I have shared in the chat box.
Yes, yes.
Okay, thank you.
So, shall we conclude here?
Yeah, sure.
Yes, yes.
Okay. Okay, sir. Thank you very much, sir. Thank you.
Yeah. Thank you.
Thank you.
Thank you.
Thank you,
Okay.
Doctor.
Thank you.
Sorry.